welcome everyone. We are having a special occasion. This is the winners colloquium of the Einstein Award for Promoting Quality in Research. Of the winners, 25. So you may wonder, we have 26. What's happening? The reason for this is that these are the winners that were selected at the very end of last year. And tomorrow we will have a big ceremony where they will receive their prizes. And there will be a next round in 2026, but that's something we will mention at the end. So my name is Ulrich Dirnagl. I'm the secretary of this award. But before we get to the winners' talks, I'm very happy to welcome Christopher Baum, welcome him in his own institute, in his own domain. Professor Baum is the chairman of the Berlin Institute of Health, and the Berlin Institute of Health not only collaborates with the Quest Center, or the rather way around, the Quest Center, which is part of the Berlin Institute of Health, collaborates with the award. But importantly and very generously, the Berlin Institute of Health sponsors this particular category of the award with 100,000 euros, and that's, I think, really great. So, Christopher, maybe you can tell us a little bit about maybe why you do this.
Right, ladies and gentlemen, it's my great pleasure to see you all here in the Berlin Institute of Health on this important occasion, to hand over then tomorrow the award and have a symposium today on the contents of your work. The topic that brings us together here is both highly timely and deeply important for the future of science and society. Around the world, research systems are reflecting on how scientific knowledge is generated, evaluated, and shared. Questions of transparency, reproducibility, reliability, research culture, and utility are now central to discussions about scientific excellence. At the BIH, I'm very proud to remark this conversation is not new. Responsible research has a long and strong tradition here in our institute, particularly through the work of the Quest Center. The Quest Center, as its name says, in the elongated form for responsible research. It has been pioneering efforts to improve the quality, transparency, and usefulness of biomedical research now for many years. The pioneering work conducted at the Quest Center is closely connected to the vision of Ulrich Dirnagl. You just saw him, who founded and built the Quest Center. Through his leadership, the center became one of the first initiatives of its kind internationally. And it's safe to say that Uli and his team helped and do still help shape global discussions on open science, research integrity, and improved research practices. Even after his retirement, which to all of us occurred too early last September, he continues to contribute his experience and passion to advancing those topics, something we are extremely grateful for. Our commitment to responsible research is also reflected in our institutional engagement at the international level. In 2022, the Berlin Institute of Health was among the first research organizations in Germany to sign the agreement of the Coalition for Advancing Research Assessment, QARA. In fact, we also, through the work of the Quest Center, even contributed to the formulation of the QARA principles. Based on those principles, we are also derived, and that's important for you to know, the key performance indicators of our own research institute. These developments also resonate strongly with current policy discussions. Responsible research and assessment reform are now firmly on the agenda of the German Council of Science and Humanities, the Wissenschaftsrat here in Germany, reflecting the growing recognition that scientific excellence must go hand in hand with integrity, transparency, and responsible use of resources. In this context, it is important to remember that scientific freedom and responsibility are not opposites. To this end, we must ensure that the principles of responsible research remain practical and applicable in everyday scientific work. It's not enough to formulate ideals. We must embed them in incentives, such as this award, infrastructures, and research culture, so that they genuinely support researchers in their daily work. The awardees we honor here today are scholars who actively advance this discussion about what good rules and practices should look like. They represent innovative proposals. They present innovative proposals for how research can be conducted and assessed in better ways, and the award itself recognizes that such new ideas can strengthen science as an institution and help it evolve. The importance of responsible research extends far beyond any single institute or country. It has become a global conversation, a global activity connecting universities, funders, and policymakers internationally. Initiatives such as Quora demonstrate that the transformation of research culture can only succeed through international collaboration. Here in Berlin, this commitment also resonates with the Berlin University Alliance, whose recent extension, just occurring yesterday evening, the approval of the next phase underscores that the city's ambition to strengthen excellent, open, and responsible science has a good place here in Berlin. Events like today play an important role in this transformation. They bring together everybody who helps us reflect on how research can best serve both scientific progress and society. And with that said, I wish you a great symposium here tonight and a great ceremony tomorrow. But now the word goes back to Uli and then to the awardees. Thank you.
Thank you, Christopher, for setting the stage, but even more importantly, for supporting responsible research at the Quest and with this award. So we're now very eager to hear from the laureates. They will present their work in around 30 minutes or so talks. We will have a question and answer session after each talk. Those of you who are joining us online, please, if you have questions or comments, you just introduce them into the chat and they will be read then to the presenters. Now, please welcome our winner for the Early Career Researcher Award. That's Maximilian Sprang. The Early Career Researcher Award is, early career researchers don't have usually a big oeuvre to show, but they have ideas and they have projects, and that's what was the basis for selecting them. In fact, through a pretty competitive process and talks in front of the jury, and so Maximilian convinced all jury members that his ideas and his project are the most interesting one. He, by profession, he's a bioinformatician and a junior group leader at the Gutenberg University in Mainz, where he also obtained his doctorate. His research focuses on quality control in multi-omics data, in generative models for DNA sequences, and in AI applications in immunology. Today, he will share how he aims to improve the quality of wet data lab work, and we're looking forward to it. Please.
All right, thanks for having me here today. And yeah, I will be talking about the project that was awarded Erring rigorously, but I will be actually talking more about the work that led me to this, which was my PhD thesis and the 4 papers that were conducted during these 3.5 years. And I have 3 little intro slides that I will quickly go through to bring us on the same page and use some words that I will be using throughout. and the first is functional genomics. So the project was focused on quality control in functional genomics, right? And functional genomics for us is basically like all the assays that give us some input on the function and regulation above the genome. So we do not look at the genes themselves as we would, for example, do with whole genome sequencing, right? But we look at, for example, the RNA with RNA sequencing to give us an information about which of these genes are transcribed at a given time in a given tissue. There are also a lot of other functional assays that we can look at. We can look at DNA methylation, for example, with bisulfite sequencing. We can look at long-range chromatin interactions, or we can look at chromatin modifications, transcription factor binding sites. And with those, we can learn something about long-range regulatory elements. So all these epigenetic regulation processes that help shape what the cell does in the end of the day. And then there are also ATAC-seq and DNase-seq, which give us an information about which parts of the genome are open in the cell at a time. And all these are also connected. So nowadays you also look at them often together and then do multi-omics, right? And in our case, we focus on these four assays, mostly actually because just they were the most abundant in the data set that we used. Quality control, I mean, I'm preaching to the choir here, I guess. So I guess everybody knows that quality control is there to ensure data is not corrupted or biased. In our case, because all these assays are sequencing assays, this actually starts already with the sequencing. And nowadays sequencing comes in FASTQ files, and the Q actually stands for quality, because they bring within these files these Phred quality scores, which are on the base call level information of how good this base call was. So how probable is it that the base that we assigned, ACTG, is actually the true base? This one we can skip. And then there is software that actually averages these scores over the whole read, over the whole sample, like FastQC, that is quite often used and that we also used in some of the projects. And then what you can also do, you can also look at your downstream analysis to get even more idea or even more information of quality. So normally what you do with all these sequencing assays is you take all these reads, right? So in these FASTQ files, there are millions of small fragments of DNA. And when they just lie around there, you cannot do a lot of stuff with them, right? So first, you need to bring them into context. And for that, you map them to a reference genome. And then, for example, count above the genes the number of reads that are there. Yeah? And when you do this, when you map them, then you will get mapping statistics. How many of these reads were actually mapped to the reference? How many of these were remained unmapped? How many could be multiply mapped, right? And this information holds a lot of information on quality too. And this is what we see here. So this is also part of one of the papers. It's basically FASTQ sequences in high and in low quality. How we came to this labeling, I will tell in the next slide. And we actually very clearly see that in the low quality regime, we have a much larger spread, and we also have very different means of this feature. And in this case, it's multiply mapped reads. And then finally, batch effects, right? So these are kind of their own niche field in quality control in functional genomics. They play a large role, especially in RNA sequencing and also already in microarrays when RNA sequencing was not around. And the umbrella term for this would be artifacts, which basically includes all these different effects that cause differences between samples that are not originating from the desired observation. So I already said that sentence quite often already at times. But what's basically it's coming down to is this, right? So what we see here are two different samples. In that case, it's single cell sequencing, single cell RNA-seq sequencing. And these two samples, they on their own generate a background signal that disturbs the biological signal. And sometimes we can correct this, and then it would look like this. So this is what we want to see. And if we do not correct for a batch effect, or if we don't know about it, then it will be looking like this. and we cannot use this data actually effectively to learn something about biology. And so, as I mentioned, a lot of this talk will be about the thesis. And here, the original aim that we started out with was just ways to explore possibilities to increase and automate the quality control of functional genomics. And in the course of that, we wrote 4 papers. The first one is SecuScorer. It's a machine learning package that allows us to score a given FASTQ file for its quality. In parallel, on the same data set with the same features, we did some statistical guidelines for quality control where we were focusing more on the features themselves, right, to learn how much quality information is actually within each of them. Then in the batch effect paper, we could show that this quality score actually concurs with batch effect, although not always. I will be going into a bit of detail here. And then finally, and this will be the paper that we will be talking most about today, the working title was QC Genes, that's why it's there. Overlooked poor quality patient samples in sequencing data and pair reproducibility of published clinically relevant data sets. And what we did here basically was show that differences in quality that confound with the biological group can actually introduce false positive differential genes. Okay, so SecuScorer. This was first authored by Steffen Albrecht, a friend of mine and former colleague, and we are still continuing to work in this direction at the moment. And what we did here was build a package of machine learning models that detect or predict low quality in functional genomics files. And we could do this by using ENCODE, right? So the ENCODE project, it's the biggest functional genomics data sets that is around. And they have very good quality standards, very good quality guidelines, also for the people and colleagues in the wet lab. And especially what they have is automated audits that assign little quality flags to the files that you upload. And then if you have a lot of warning flags, so potential problems with quality, then the file will go to actual humans. So it's human labeled data. So everybody that does machine learning love this, yeah? And they will then finally decide, is a file released, so good or good enough, or revoked. And now comes the clue, the revoked files, even though they are of low quality, right? They still are publicly available. You can still download them. You can still use them for biological analysis or, like us, to build a classifier for quality. And so in the dataset, we have RNA-seq, DNA-seq, and ChIP-seq data. And we could show that the algorithm generalizes to ATAC-seq data. We could also show that between human and mouse data, we see generalization. And so for all this downstream work, we actually use what we call the general model, which has all the data, right? Again, from the machine learning side of the things, you always want to cram in as much data as you can to learn as much as you can, or the machine. And what we basically do is first we derive our features, and for this we use software that is already established in the field. So this first that we call raw is actually FastQC, right? So the most used quality control software for sequencing data. And we just took the output of FastQC, which basically gives you flags, and made an ordinal scale out of it to put it into the machine. And then here we have the mapping statistics that I just mentioned, right? So we map the sequence to a reference genome, and then we look at the mapping statistics and use those as input for the machine. And then finally, with these mapped reads, we look at the reads that are mapped into genomic locations, right? So introns, exons, distal intergenic, 3' UTR, stuff like that. And then in bins around the transcription start site averaged over the whole genome. And with this information, SecuScorer can actually quite well predict the quality of a given file. And this is the take-home message, right? So we provide software that enables automated quality prediction. You should not rely on it alone, obviously, but it can help a lot. And then, here's also the picture missing, but okay. I will be going quickly over this anyway. So this is from the second paper where we were looking for statistical guidelines, right? And here we had the same data set, so we also based it on the flags of ENCODE, and then we divided it by all this metadata that we had. So organism, biosample, tissue, for the ChIP-seq even protein target and antibody, so that we have what we call experiment-sized data sets. And they basically ranged from an N of 8, which is quite small, but which you can find, especially in smaller labs, right? And to roughly 60 was, I think, the biggest one. And there we could actually show by simulating decision-making just with decision tree algorithm, right, that you just need like 3 to 4 of these features to divide all these data sets into high and low quality. But what we saw here was that the thresholds that you would use for this are not always the same. So they are not global, right? So they are more like a fuzzy thing. And this is also reflected in Secuscorer's score. It will not give you like the ultimate score of quality, but you always need to see it in context, right? So if you have a big experiment, for example, and another big experiment, there will be differences in the quality score on average, let's call it like that, right? So there will be a given fuzziness between experiments, and this is connected to the next point, and at the same time also allows us to look at the next point, which is the batch effects, right? So the batch effects I mentioned already, they are quite important, and we asked ourselves, would our quality score be able to detect batch effects, right? And here we see a case where it very clearly works, right? So on the x-axis you see the batch, on the y-axis you see p low quality probability, and there's a very clear difference again in mean and distribution. And also sometimes you can use it for correction of a batch effect. So this is one of the smaller data sets, right? So, and here we just worked with, let's call it random data sets from Geo. The only criteria that we had is that the batch information was given, right? Because we have to test against something. And here we can actually see on the x-axis the first principal component. So it holds the most information basically when you embed the data. And this is basically just the batch, right? So the batch signal is here actually stronger than the group signal. However, when you correct it with either the actual batch information as well as with p-load, then you see that you get rid of this batch effect. There is obviously a lot of software that tries also to do batch effect correction, right? And then it's still like a growing field. Like I reviewed like 3 works with new packages that do this in the last 1.5 years or so. So there's still a lot of work to be done. And this is also owed to this fuzziness that I already mentioned, right? Because here, for example, in this example, I would call this package the state of the art. It's called the surrogate variable analysis, SVA. And here it performs much better than us, right? So it basically performs as good as the original batch information. However, there were also cases where it was the other way around. And this was most of the time the case when SVA overcorrected. So it's cut also on the biology, not only on the technical stuff. So take-home messages here. The features on their own are informative of quality and can also be used in small subsets. So you don't always need the full bulk. And then quality information is connected with batch information. Okay, and then the next and last paper where we were looking into these quality imbalances that I mentioned at the start. So first, a few words for the quality imbalance. How would we measure that? So 0 and 1 here is just the group. And group here means in 40 data sets in Geo, disease was healthy in actual patient data, right? So this is all patient samples of disease and a control, either adjacent normal or sometimes even really volunteers, right, that were actually healthy. And we would then assign to each group just 0 and 1, and we would not care for healthy or disease, which is which, because we just look for the absolute value of the correlation of the zeros and ones against our quality score. And with that, we basically set a cutoff at 0.3, just because the 0.3 is often used in biology as an effect size that is seen as, like, say, it's strong enough, right? And we just picked this randomly at the start and it worked quite well, so we stick with it. And when you use this 0.3 as a cutoff, actually 30% of the data sets that we were looking at had a quality imbalance above that. So the natural next question is, does that impact the downstream analysis? If it wouldn't, right, we wouldn't need to care. However, the answer, I don't know. The answer is yes. You cannot see it, but what you would see, is on the x-axis the number of samples of all these data sets, and on the y-axis again our score. And when you compare the samples with a high quality imbalance, right, then the slope of n versus the number of differential genes is higher, stronger. Slope is stronger, right? And with a rising number of n, you would expect that to an extent, right? Because the more samples you have, the stronger the statistical tests get, in statistical power that you use to get these differential genes. Yeah. However, since the slope is so different, we actually see that this quality imbalance introduces false positives. Yeah. And now one could ask, I hope the other talk we will be able to see, but so now one could ask, it is how it is, no worries. Perhaps you just got lucky or unlucky, depending on the point of view, right? Perhaps all these high quality imbalance data sets are cancer, right? And all these others are illnesses where you expect a lower number of differential genes, right? So that could be perhaps there are psoriasis, inflammatory skin disease, or Alzheimer's or something like that, because all this was in this data set. It was not just cancer, it was very heterogeneous. And so we did a little, we did a little, let's call it simulation experiment. We took the biggest data sets that we had in our data set collection, and we ordered in each group healthy versus unhealthy, right? Low to high quality by our score. And then we sampled out of this top and out of this bottom always 10 samples at random. And then we would put together either low and low quality and high quality. And that would give us data sets with low quality imbalance, right? Because both groups are similar. And then we just cross-paired them here. So low to high and high to low to get data sets with high quality imbalance. So, and this brings us to this, and this time on the x-axis is not n anymore because n is always 20 now. But on the x-axis we have the quality imbalance, so this correlation score that we assign, and on the y-axis again the number of differential genes, and we see a very nice linear relationship here between these two things. So again, this points at false positive differential genes being introduced here. How much time do I have left? 15 minutes. Okay, nice. That's very good. So, and that brings us to another question in the narrative of the paper. But to be honest, this was the original question that brought us to this work, right? Because it was also meant as a bit of validation if SecuScorer actually works, because parts of this is expected, and I will come to which parts. So the question here is, are there correlations between quality and gene expression, right? And let's focus here on this part for now, both because of time and because this part is actually the more easy to explain. These are the genes that are correlating with low quality. So that means they are high expressed in samples that have lower quality than the others. And what we see here already in this is just the top 20, are mitochondrial genes, right here. There's also oxidative phosphorylation genes, right? So, and this is known also already since the microarray. So mitochondrial and ribosomal RNAs are known to be enriched in samples with low quality. And this is rooted in their biology, actually. So, and when you do a pathway enrichment of these lists of genes, then you will also see this, right? You will get here ribosomal proteins. This is quite small, so sorry for that. You will have oxidative, phosphorylation, and you will have housekeeping genes, so mitochondrial genes again. And like I said, so this was what we expected, and actually showed us that our work kind of made sense. And these genes, they have been used as an indication of low quality since the microarrays, and are also, for example, now used in single cell RNA. So single cell RNA, especially mitochondrial RNAs, are used as a literal cutoff for quality. So depending on context, between 10 and 20%, you just cut off and throw out the cells that are above that. However, we also saw some stuff that we did not expect at all. So what you see here are Parkinson's disease, Huntington's disease, and Alzheimer's disease. And these pathways, they would always show up when you do enrichment of these genes that are correlating with low quality. And we do not know why. So in case you wanted to ask, I'm sorry. But I bring that up because one of the reviewers of the paper actually asked the same question. He was like, we also see this in our lab when we have low quality samples, that these pathways pop up, but we do not work in neurodegenerative diseases. So why is that? So sadly, we could not answer, but he still accepted it very nicely. But this is definitely something that I also want to follow up in the future. Okay, so to the conclusion, quality imbalances between target groups lead to reproducibility problems, especially to false positives and differential analysis. And quality imbalances concur with certain expression patterns. And now this brings me to the project which brought me here, Erring rigorously. And this is basically the natural extension of the in silico work that we did. So now we bring it back to the wet lab, and we want to try to learn something about these, the impact of different errors on quality, as well as on batch effects, right? So, or rather on the sources of batch effects, to hopefully then be able to correct them better, right? Or even don't make these errors from the start, right? Perhaps we can also improve the wet lab protocols. I don't know. We will see. And here, really, just the game is basically to systematically and deliberately introduce technical perturbations or errors, right? So it's not really errors then anymore when you do it by yourself. Then what we want to do is basically use our tool, right? So we have the normal experiment, we use our tool, we get a low probability of low quality, and then we introduce errors of different kinds, and we would expect that also the score differs, because sometimes an error will be having stronger effects than others. And to do this, we start off in cell culture first, because we have most control of what we are doing there, and second, because ethically it's basically the only possible way to do this, right? Because you cannot kill animals to do error research. And so this is actually a bit outdated here. We don't do a knockout versus wild type, but we do a treatment versus the control here. And we use a cancer cell line. And we opted for that because the partners in the wet lab that do this for us have also old data that we can then also bring in to have like an additional batch effect that is really long time ago. But we will also, and this is what this empty line is here, we will also introduce our own batch effect by having two different handlers. And then what we will do is we will change the cell passage, change the cell culture density. So we will directly interfere basically with the cell culture itself by either passaging and splitting it more often. And by more often, I mean really often, like 60 times. And then we will interfere with the culture density by just seeding more cells, right? So when you have your little flask, you put the cells in after a split. That means split always means you take some out from the culture before, and normally you would put in like 10% to have them, give them space to grow again. And in that case, we could go with 20, we could go with 30, and we would basically have multiple steps to have different conditions. And then finally, because we are looking at RNA-seq, we can also interfere with the RNA itself directly, and we will do this by enzymatic digest. And here again, we will have steps. We have one digest of a certain time and one digest of the double time. And then downstream, after sequencing, what we will do, we will use our quality scoring tool. We will do classical downstream analysis, differential expression analysis, clustering, to see how do these changes interfere with our perceived biology, right? And also, can our quality score software actually catch these scores? And then can we, these errors, I'm sorry. And then can we use this score potentially to correct for these errors? The expected outcomes here are mostly the data set, right? And an annotated data set like this could potentially be a real gold standard for all these software tools that try to correct batch effects, right? And as I mentioned, this is still a lot of ongoing research, and there's still ongoing need also for them because there is no size fits all yet. Potentially, we could improve our own software, right? So if we see the software doesn't pick up any of these errors, obviously that would not be so nice, but then perhaps we would want to try to build it better. And then finally, and this is like the vision outside, is this could be a first step towards data-driven guidelines for sequencing, right? Obviously, the amount of sequencing experiments that we can run within this project are not enough for this, right? But potentially, if we see some exciting results, then we can bring in some partners, ideally around the world, and then decide on a biological system that we want to work on, and then have something like a batch ring experiment where we have multiple experiments running in multiple labs, and then introduce different errors at different places, etc. There is some other related ongoing work that also goes in this direction. I have shared. Okay, I have a shared PhD student, Johannes Wolter. The picture is not very good, but he's actually very handsome now. And what he's working on is, can we learn representations for batch effects or other technical artifacts in the data, and this time not supervised, right? So what we did before was supervised learning. We had labeled data, and here we want to use variational autoencoders to potentially learn representations for these artifacts without having the labels. And then, okay, there's another picture missing. So there would be a picture of single-cell RNA-seq here. And here we ask the question, can we use biological information to combat batch effects? And the biological information in this case that we are using is alternative polyadenylation patterns. I will not introduce the concept here now because this would take too much time, but the gist of it is basically you can compute this with all RNA-seq data that is around. You just need normal BAM files, right? And then you can compute alternative polyadenylation profiles. And they give you normally just information on regulation, right? Because it's one of the many tools that a cell has for regulation. However, the patterns that you see there, they are more robust against batch effects than the normal RNA-seq counts. And we want to use this information also to correct for batch effects. And this is a work in collaboration with Mert Cihan, also a friend of mine and former colleague. And this is also still ongoing. So this is a little list of all these projects that are ongoing. Erring rigorously just started. The experiments are running right now in the lab of Julian König that you see here on the side. Very nice of him to take that on because I cannot do it. I'm just a bioinformatician, right? And then we have the PhD thesis of Johannes, who is working on the autoencoders, and on this upper project, he will take it over from Mert because he will be changing positions soon. There's a bachelor thesis going on, which is focused on fixed and non-fixed tissue, right? So paraffin-fixed tissues are still used a lot for histopathology, and they can also be used for RNA-seq. And then we are looking to integrate this kind of data. And then we have two papers in submission, and also Steffen Albrecht, who was mentioned at the start, who is the first author of SecuScore, is also again part of these. And one is just focused on the dataset publication for quality features, and the other is an extension on SecuScore using the ENCODE log list. So, and with that, I'm done. Big thanks to the CBDM group where I did my doctor with Miguel Andrade, this guy here, very nice, I can recommend. And also thanks for my current lab, Johannes Meyer is the boss there. And if there are any questions, I'm happy to answer. Thanks.
Thank you very much. Perfect timing, by the way. We have 6 minutes or so for discussion. May I just start with something that puzzled me? We learned in the what you're planning part that obviously these batch effects are a black box, right? And you're trying to sort of find some elements of the black boxes. But earlier on, you said that you can correct for those batch effects, and that's kind of puzzling for me.
Yeah, there are 2 ways how you can pull that off. So one way is you already know it, right? So there are basically surrogates that you can just use for this. So when you actually know the batch, right? So you did it on one day, you did the experiments on the other day, then you have 2 batches, and you can use this number or this information to correct, right? Like the easiest way to do this is to basically do z-scoring on both of them on their own, and then they are closer to each other because you scale them, right? And then there are more modern methods that use linear models to also take into account gene effects. Some genes are longer, some genes are shorter, and will generate different kind of noise, right? And so they are a bit more sophisticated. And then there's also, and this is the tool that I call the state of the art, this SVA. And what they do is basically they take the biological information that you normally should always have, right? So healthy versus healthy versus disease. And they use a linear model to get rid of the signal. And then they have what they call a residual matrix. And then they just use a PCA-like decomposition to get these representations.
I don't know about the Ulrich, do you see the chat? Is there something in the chat? Anyone in the audience? Evgeny.
Yeah, I would like to know whether you think that repositories like GO could or should do something about it.
Did those in the online hear it? So the question was whether repositories would help.
Yeah, so especially GEO is a very good thing to mention here, because we work with it for the batch effect and also for the quality imbalance work, right? And we had a lot of problems to find enough data sets that had well enough metadata to even do this. So one thing that should go first in these repositories is to actually try to bring the people, to push the people to actually annotate their data well. That already would help a lot, because with this annotation, sometimes you can also find information that is a good surrogate for batch information, for example, like the date on which an experiment was done, right? But I mean, this kind of information, sometimes you will just not have it, or it will not be very clear, but information like sex of a patient, information like age, information like genotype of a mouse should always be there in the first place. And this should also be shared together with the data, and sadly, it's often not the case. So this would be the first thing that they should do. And then, obviously, if they can implement quality audits like ENCODE does, then that would also be very helpful. But there's also a lot of infrastructural need then. So I don't know if that would be justifiable.
I just wonder, looking at your batch effect correction, shouldn't we not more focus on reducing the batch effect by experimental practice in the first place?
100%.
And then to get rid of the batch effect as good as we can by the quality of our experimental procedure.
Yeah, 100%. However, to an extent, so we cannot get rid of it completely, I believe, right? Because biology by nature is noisy, and a biological system, to bring it into such a stratified way that there will be no technical effects that influence it is kind of hard, I think. So I don't think it's completely, we will be able to completely get rid of it, but it's definitely the way to go to try to minimize it, and also to try to then annotate it well with the metadata. Right?
And can you use internal standards to identify the batch effect even better?
Yes, 100%. So like we are planning a little project at the moment, not so little project at the moment, with a lot of partners, and there is actually part of the Z project there to set up standards like that and to keep as much information from the lab as possible, right? To have a strong metadata and data annotation background.
Yeah, so that was super interesting because the batch effects not only occur, of course, in genetics or in sequencing, they occur like in every researcher's life. Like you have this between MRI machines, you have it between labs, you have it between days, you have it between experimenters. So one interesting thing is, so two questions basically. The first one is, how does the heterogeneity that you detect compare to the heterogeneity of other batch effects, now fields? And the other question is, now that you have a kind of quality control measure, why not just discard those low quality samples?
Okay, so first question is very hard. I don't know if I have a comprehensive answer for that. But so what I know is like mass spectrometry proteomics, for example, and there the batch effects are kind of similar. However, the sources for these batch effects can be very different. So in proteomics, for example, there is a much stronger difference already between the machines that have been used, which we don't see as much in sequencing, for example. But then what we would see in both is, for example, differences between handlers, right? And for the second question about just discarding the low quality data, and I would say no, because there is still biology in that data, right? Or it's like it's context dependent. So if you have a small data set, right, of let's say 4 against 4, then perhaps it would be just better to discard this one outlier if it's like one quality outlier. But if you have a big data set, right, of I don't know 100 samples, and there you have like a distribution of quality over the whole thing, then it's I think a better idea to take this information and put it into the model that you use to analyze the data. And in general, I'm always pro keeping data. So also, please, if you have low quality data, upload it still and just annotate it well, because then we can improve the software, right? So.
Okay, thank you very much. Congratulations again. Winning the award, and we are moving to the next award category, which is the institutional one, and that's also endowed with 100,000 euros that are generously provided by the Württemberg Wissenschaft, and this in partnership with the Berlin Senate. This is also true, by the way, for the individual award, which is also sponsored by those two entities. Now, the 2025 award, it went to the Reproducibility Initiative, the Brazilian Reproducibility Initiative, which is a nationwide effort to systematically evaluate research results in laboratory biology. And by the way, the largest coordinated replication effort in the field worldwide. Now, I'm happy to welcome Olavo Amaral, who is an old friend of Quest, and he is the coordinator of this impressive grassroots initiative. Besides that, his role is professor of medical biochemistry at the Federal University of Rio de Janeiro, and we're looking forward to your talk. And congratulations.
Thank you. It's a big honor for me to be here representing the Brazilian Reproducibility Initiative. It's been quite a long road. It's always a long road from Rio to Berlin, but this one is a rather long one. I mean, you can judge by my hair color rather changing from the start of the project to today. But I mean, this is really kind of like a landmark for us. We have applied for the Einstein Award, I think, since it started in 2021. We've seen multiple other projects which have very much inspired our own being awarded in former years. So this is kind of something special for us. We started off in 2018, concerned about reproducibility in lab biology. I think the discussions on that have been picking up for a while in the literature international as well, but we did have actually little empirical data. I think we still have very little empirical data on how reproducible or how easy it is to replicate an experiment in lab biology. We did have some numbers from pharma companies. We have some small-scale efforts within specific fields of science, but we actually did not know that much about numbers of how much can you reproduce. And more than that, working in Brazil, I wasn't really sure how much this was actually applicable to my own context. I mean, and I was doing a lot of reproducibility advocacy like around 10 years ago, and I had very little data in my hands to argue with people in institutions or funders like how relevant this is to our particular context. Context there is Brazilian science grew very fast from the 1990s onwards with the consolidation of the Brazilian graduate education system. A lot of people came into the system, a lot of metrics came around in terms of evaluate and distribute money around those people. And that was concerning. I mean, can you keep up that rate of growth with like actually producing reliable science? And that was a big question to me and to other people as well. So we tried to produce data on this, so like how is reproducibility doing as science rapidly expands, and we came up with this project of the Brazilian Reproducibility Initiative, which is a multi-center effort by Brazilian researchers to estimate the replicability of experiments with some common methods in Brazilian biomedical science. We've been covered a lot in many different venues over the year. I do think we're still the largest replication project in the world in terms of numbers of labs or numbers of experiment articles involved. So we're very much a crowdsourced effort. We put up open calls for any lab in the country who work with particular methods to join. We're actually very surprisingly successful at having interest. So almost 100 labs signed up to participate in the project. We were able to have 75 of them join the project at some point depending on method, and 56 contributed this one experiment in the end. So it's big and very much distributed over Brazil. And our initial goal was to replicate 6 individual experiments from Brazilian articles using 3 common methods, which are the MTT assay, which is an assay to measure, widely used assay to measure cell viability in culture, RT-PCR, which is a very standard technique to measure the expression of specific genes in tissue or culture, and the elevated plasmase, which is a task to measure anxiety in rodents. So, and the reason for this is like these are very common methods. We had a lot of labs working in this that could replicate experiments. And so any experiment with these methods and using cell lines, rats or mice as biological models was eligible. And we took these experiments from a 20-year random sample of articles in life sciences journals with at least half of the authors, including the corresponding one based in Brazil. We full text mined these articles for experiments with these particular methods and sent them to the labs to judge whether we could actually replicate them. And so it's pretty much a representative sample of Brazilian articles, perhaps enriched in simple and cheap experiments, which we could do. And when we finally got enough of them, each experiment got sent for application by 3 different labs who were blinded to the original source of the experiment and the results. All they got was a transcription of the methods, including the gaps in the methods, which were sent as questions that the lab had to answer. Like, if we don't know how this was done, how do you plan to do this? This leads to 3 non-identical replications. So each lab independently fills in these gaps and adapts things if they judge necessary. And it comes up to be kind of like a naturalistic estimate of 3 labs doing their best to replicate an experiment from the published literature based on what's available. We try to follow, as other replication projects, a very confirmatory mindset. So every protocol got internally peer-reviewed after it gets fulfilled by at least 2 people. They were pre-registered with embargo at the Open Science Framework. Data collection was done by very obsessive, extensive templates that not only ask for data, but like for very many steps, so we could audit the methods as well. And we predefined an analysis plan to assess replication rates and predictors. That said, as much as you can review the protocol, people break their protocols all the times. So we had to institute a post-experiment validation step in which we took these data forms, reviewed them. The coordinating team reviewed the data collection forms, identified protocol deviations or technical issues, and sent these without the results. So it was like results line validation. We sent a summary of the deviations on the method to a validation committee of 3 people who had to judge whether this was still a direct replication, a valid replication. If anybody raised concerns, we discuss this live and decide whether to include or not the experiment in the primary analysis. And after we did all this quantitative analysis of experiments and actually published this overarching analysis, we also took a qualitative assessment step in which we took all the labs that were involved in the replication of a particular experiment sent them the results of every other lab. We looked at it together. We discussed whether they thought their finding was replicated, was not. If not, why? And this actually led us to identify a whole new round of data issues which we had previously overlooked, and validation decisions which had to do again, because if you talk to people, you just like learn a lot more than if you actually look at the data collection form. So big lesson. Project numbers cost around 200,000 euro. I mean, it's quite expensive by Brazilian standards, not so much for European ones perhaps. Took us 7 years of work. That's still more than expected, largely due to the pandemic. As we were starting, pretty much every lab closed down. We were like stuck for 2 years waiting for things to resume to normality. Over 200 offers, almost 100,000 purchases, almost 400 coordinating team meetings. This is the one that impresses me most. And a lot of steps to describe. And this is what we end up with. So we started out trying to replicate 60 experiments. 3 replications, each 180 replications. We're able to get in this one data point for like 143 of them, which is actually quite good. That's over 80%. Of course, some got canceled because we could not get materials or because labs left or did not finish in time. But out of these, we lose a lot of our sample because of the validation process. So like 53 out of 143, or like over a third of replications had some issue that if we put it out of the primary analysis, either because people deviated from protocols or because there was not enough biological variability between experimental units, or sample size was insufficient, documentation was not appropriate, results were known. So like we do lose a lot in this validation process because things go wrong all the time. And we end up with 90 replications for 45 experiments, which means that most experiments will have 2 replications. Some will have 3, some will have 1, but this is what we work with in the primary analysis. And so it's 45 experiments with, on average, 2 replications each. And these are our successful replication rates. So we have 5 pre-registered replication criteria. And our replication rates, depending on the criteria, varies between 20% and 44%. So 20% below is 20% experiments are significant, statistically significant and regular regressions in the same direction. In 22% times, most of the replications are significant. In 29% of times, the replications effect is within the original confidence interval. In 42% of times, at least half the labs consider the result subjectively replicated using neural standards. And 44% of the times, the original effect is within the replication's prediction interval. That's how, and this is quite an optimistic criterion. I mean, if you get a lot of variability between replications, pretty much anything fits, right? So, but this is what we get. In terms of the effect sizes are smaller in replications. This is pretty standard for any replication project, I think. There is a correlation between the original effect and the replication one, but it's actually very weak when you remove the super outlier that has humongous effects on both replication and original. It's kind of like a cloud of points. And there's also a lot of variability between replications, even though people are not biased towards any particular results. They're just like trying to do the experiment following the protocol. You do get, so this is in dark, you have the original effect size. The light dots are the replications. And you can see that sometimes you have very, very, very big disagreements between replications themselves. That said, this variability is still smaller than the one between original studies and replications. So there seems to be some amount of reproducibility that has to do with something in the literature that is still larger. Like the variability between what's published and what we got is still larger than the one between our own results. There are some caveats here. Well, one big caveat is like either due to missing replications that did not get done, or to variability being much larger in the replication. So the coefficients of variation are typically larger in the replication than in the original. This is particularly true for the PCR experiments, where we get like much greater variability between experimental units than what is reported in the original articles. This means that some replications ended up underpowered to detect the original relative difference because the application was just a lot larger. We can back calculate power using the original relative difference and the observed variability in the replication. And you can see that although most experiments do keep adequate power, you do have some that are strikingly underpowered to detect the effect that we were looking for. So, I mean, you can filter those out and redo the analysis, and then you get replication rates between 26% and 38%. They kind of converge. You can also do all kinds of different analysis sets. You can just like not consider the validation process. And curiously, that does not make that much of a difference in the replication rates. Or you can be stricter and use only experiments with two replications, three replications. This does lead to slightly higher replication rates. If you just like do a multiverse analysis, including all options of criteria and selection of experiments and some other statistical particularities, you do get a median replication rate of 25%, which is not high. It is not super outside of what has been reported in other fields and other parts of the world as well. So I mean, it's I don't think replicability efforts in biology tend to find this replication rates between below 50%. We also looked at predictions of predictors of replication success in the sample. In terms of t-values, like predicting the replication t-value, the measure of evidence strength, by far the best predictor were researcher predictions before we started out. So we had a cohort of 70 researchers trying to predict what would replicate, what would not. They vastly overestimated replicability, but they had some accuracy in terms of like selecting the ones that actually ended up replicated. The other factor, but it's a weaker correlation. I mean, if the original last author had published a lot in the previous 5 years, it actually correlates with lower replicability. It might make sense, but again, this is very tentative. I mean, there's like a lot of predictors, so I'll call this quite exploratory. In terms of effect size, predicting effect size decrease, the difference between the effect size and the original replication, the method and experimental model are the ones that matter most. So MTT experiments and cell line experiments in general have lower effect size decreases than elevated plasmase experiments and animal experiments as a whole, which on average were have lower replication around the board. This was published as a preprint in 2025, made some headlines both in the Brazilian media and in the scientific literature worldwide, and then had to be republished in 2026 because it started out the quality of assessment pattern. Just like realized so many small and some large errors actually got passed because I mean all this asynchronous things, but like yeah, you send us the data, we correct, we send it back. Things get lost along the way. I mean, it's very easy to get to misunderstanding. So we put up a warning note on the preprint and actually revised it in 2026. And now I feel much more confident that we actually get the data right. But this was really like just one of the instances where we realized that, I mean, the burden of irreproducibility was on us. I mean, it was us messing up another problem in the literature. That happened multiple times. But the beauty here is that it doesn't really matter much. I mean, if it's like Brazilian researchers replicating the work of Brazilian researchers, like if the problem is in the original article or if it's the problem on us, in the end, it's our problem anyway, right? So I mean, it's something that has to be fixed. We're kind of like looking at ourselves in the mirror. The populations of labs are actually very, very similar. I mean, between the original authors and the replicators. So I mean, whoever's right doesn't really matter. I mean, we have trouble replicating wrong results, and that is our problem, right? And actually, at some point, we realized that the real rich data set here was not something we learned from the literature, it was what we learned from our own replication. I mean, if we cannot replicate an experiment in the literature, okay, yeah, we cannot replicate it, but you don't really know what happened. But like when we messed up, we know very well why we messed up. And we have like a very, very big log of errors and problems and protocol deviations to look upon it. And this is actually what we really, like most of what we learned has come from our own mistakes and problems. And when we realized that, we started a self-assessment project with the labs, evaluating replication. So like, do you agree with validation with the decision? Why did you deviate from the protocol? Could you have prevented this? Would you have done something differently if you started over? What you learned? So we do have a lot of data on this. Some of it is in the paper. I won't go through the actual data, but I'll just go through a few takeaways of what we learned with actually, like, again, very much by looking at our mistakes. One thing is that I think a lot of the problem here comes down to like lack of standardized terminology and common language between labs. I mean, for very basic things in terms of experimental design, people have very established ways of working, and they're not always the same. So like the paradigmatic example here is the experimental unit in cell line experiments. So like if you have an N of 3, what is 3? Is it 3 different passages of the same cell line? Or you run 3 cultures in parallel and like do it all on the same day? Or you take one culture and like people have different ways of doing this. I won't get into what's best with what, and there's pros and cons. But like they usually do it their own way, like the other lab does it their own way. Nobody really describes it. It's very hard to assess in a published paper. And people have a hard time even describing. Like, we had to go back and forth with emails just to understand what was happening. to a lesser degree, I think the same holds for things like the naming of positive and negative controls, experimental blanks. People use this term for different things. And it's very easy to, we probably were clear, like what should be or not be. People interpreted this in like 4 or 5 different ways. They had a hard time explaining it to us. So I think like just like having standardized terminology is like a low-hanging fruit to improve communication and reproducibility. Another lesson is that we try to pre-register protocols and be like very strict like with this direct replication thing, but it's actually very hard in biology to do it in advance before you just even do pilot experiments, because biology works in its own way. So like sometimes you can try to follow the protocol as a recipe and like the model just doesn't work as planned. Or sometimes you can even follow like grow your cells for 1, 3, or 5 days to get 50%, 80%, or 100% growth. I mean, sometimes you grow it for 3 days and get 60% or 90% because I mean, whatever conditions change. So it's very hard to follow protocols strictly. Pre-registering ahead of piloting may be an unnecessary burden because I mean, it's just like people will end up deviating anyway, right? So, and again, what is the best potential? It's a judgment call here. I mean, should you keep the time constant? Should you keep the confidence constant? I mean, labs have different interpretation of this. So it's actually like the concept of like, yeah, you just follow the protocol and everything's going to turn out right. Not really sure that actually applies. I mean, sometimes the best replication may not be following this strictly, but it may be just getting the experimental model to work as planned. Another thing, I think this is a big one. I think error is underrated in the reproducibility debate. I mean, there's all kinds of reasons for irreproducibility, which go from like real heterogeneity between labs, like conditions changing, to random variation, to honest error by humans, to bias in questionable research practices, to fraud, fabrication, etc. On a scale of like the worst to the less bad. And I think people tend to look at different points in the spectrum. So like if you talk to biologists, they really are big on like, yeah, little protocol variations or changing conditions. Statisticians, of course, will talk about random variation and sufficient power. Psychologists are really into bias and questionable research practices. People look at fabrication. But I don't think error has enough advocates, and I think error is very common. Like silly human error is very common. Like people just forgot to put a sign in the formula. They calculated the results. It goes the other way around. Or they copy pasted something from one spreadsheet to the next and like reverse the group labels, or 20 millimolar become 20 micromolar when something around, and something you're using like 1,000 times less drug than you need to. These are actually real examples. I mean, you have all these kinds of like very, very, very small errors which make a very big difference. They were widespread in our sample. I think we managed to catch them because we actually included very extensive audit of the data collection forms. But I think these are very prevalent in the literature as well. I think we underestimate just the potential for human error if you don't have very tight quality control, which I don't think we usually have in the lab setting. So really, I think the conclusion here, we actually need much better standards, both for data management. So like labs record as much as you try to like provide them with standardized data, but people just like edited the spreadsheets, like put their results in their own way, or they had like some, this is actually on this other spreadsheet that is in there's colors. And it's complicated. So like I think the average biology lab is not very good at data management, and we're actually not very good at proactive quality control. So we put a lot of effort in reviewing the protocols. We put a lot of effort in the end in reviewing the data, but like there's process, right? And during the process, we had like 56 labs in different parts of the country. Of course, we could not be following this strictly, and I do think quality control is needed in terms of like reviewing things at every step. To make sure that things go right. I don't think we're very good at it. I mean, compared to like industry or other human endeavors, I do not think quality control is a very well-developed science within the academic environment. Like because I think, and this has to do with a human aspect of labs as well. So like academic labs are typically small, like a PI and a bunch of students and one postdoc or something. They're not super robust to contingencies. A lot of the project's memories deposited in specific individuals. So people leave, teams change, and this happened a lot because of the pandemic. It's a mess to actually figure out what had been done previously. A lot of the sensitive steps are left in the hands of a single person, frequently a student. I think this also explains why you have a lot of potential for error. And we improvise a lot, right? I mean, and Brazilians are very proud of improvising. Like, well, we do a lot of research with like little resources. But improvisation at some point takes a toll. We have to interesting anecdote. So this is a lab that was submitted data. And if you look at the dates of the experiments, like everything is a Saturday, Sunday, or holiday. Like this is somebody working as a side project from whatever they were doing the regular time. We interviewed people who say, yeah, I did all the experiments between like 5 o'clock in the morning and 8 o'clock in the morning, because then later I had to work as a personal trainer because like the master students scholarships in Brazil was meager and I have a kid to feed and stuff. I mean, and so I think a lot of the human error thing comes down to like human conditions, and this is frequently not ideal, especially in Brazil. Of course, this I think explains a lot in terms of why things don't always go right. And the other aspect of the human thing is we're not very good at coordinating. I mean, we're not used to this. Communicating with like 200 people and coordinating 200 people for me is like a completely. I was never trained for this. It's very hard. You do need a lot of hierarchical coordination, and it's super hard to actually learn. We learned a lot, but I mean, it's hard to do, and it's weird for the labs as well, right? So like people are not used to being supervised from somebody from outside, especially like somebody who actually lacks expertise in what they're doing. Because I wasn't an expert in like PCR or NPT experiments. And so it's hard to standardize practice. I mean, you can try to like, yeah, please follow this instructor. People will revert to their traditional ways of working. I mean, we used to collaborate and like, yeah, I do my thing, you do your thing, we join the data together, put up a paper. But like somebody from outside, like telling me what to do is like, So it's hard to standardize procedures, and asynchronous communication only goes part of the way in terms of like getting people together. I mean, people are very used to like talking, and so we try to do the project in this very decentralized way. So we send instructions, send a data, we send a form, you correct, but like a lot of things get tested. So like if you wanted this to work, I think you have to have very close supervisions and like actual like real life meetings with people for this to happen. And to me, the metaphor here is really like you can put a lot of garage bands together, but that does not make an orchestra. I mean, and academic labs are very much like garage bands, and there's nothing wrong with this. I mean, I think you do have a role for very free, exploratory, not very supervised research. I think a lot of what's good in science comes from that. So I have nothing against garage bands. So here's me playing one actually. But sometimes you have to play like an orchestra, and I don't think we necessarily, it doesn't exactly scale up. I mean, it just like can put a lot of people who are used to playing like this and get them playing like this, because these are different. These are very different things, and I think we're very much skewed towards exploration. And I do think that perhaps I think there's all this meta science discussion of like science slowing down. We had like a massive growth in numbers of science and like resources invested in science over the 20th century, and like yeah, science grew, but like perhaps not as much. I don't have to position this, but like a lot of people talk about this. I think this has to do with scaling up poorly. I think we have like a lot of human capital being poured into science, but we didn't really develop the structures to actually make the best use of these people. I mean, we went from like, yeah, a few people doing experiments like this very handed way to a lot of people doing experiments in this little handicraft way as well. So I think we could scale up better. So I mean, this is just an impression, but I do think that it could help. And to me, the last years, like confirmatory research as we had set out to produce does require better orchestration of efforts. A lot of our spin-offs from the initiative have gone in that direction, trying to get people to collaborate in more efficient fashion. We do have Brisa, which is during the pandemic, people were like stuck with no labs and like, yeah, let's do systematic reviews. So we created this collaboration to actually train preclinical researchers to do systematic reviews and do hands-on work. We now hold periodic public calls for new members. People go through an online course, which is partly open, and get engaged in collaborative projects of their interest. And this has been running for 5 or 6 years now as a full-fledged collaboration, which sprung originally from the initiative. And the other thing that sprung from the initiative is the Brazilian Reproducibility Network, which was something we, from very early on in the project, we wanted to do something like this. I mean, realized we had the human capital and the visibility to be more than a replication project. Shortly after we launched, the UK Reproducibility Network was launched in 2018. This kind of like became a template for reproducibility networks in a lot of countries, including Germany. We launched our own in 2023. Now has independent fundings, 5 management categories, many disciplines, and it's been growing and should carry on the legacy from the initiative. So it's a community-led organization to promote open reproducible research practice in Brazil, beyond the replication project itself, through community building, training, meta research, and activism, has almost 100 members at this moment. It's funded by 2 philanthropic entities and has 2 full-time hired executive directors and elected coordination. Does a lot of stuff. I won't have time to go through this, but we do events and debates on reproducibility and open science across Brazil. We do recommendations for promotion of reproducible research by different stakeholders, by journals, by institutions. Most notably, we have worked with CAPES, which is the agency that funds and evaluates graduate programs in Brazil, to help them consider open reproducible practices within their evaluation. They do now have a dimension for open science, which every graduate program should do. We're now trying to work with the programs to help them conform to this. It's a slow run. I mean, this changes very slowly. There's a lot of inertia, but I do think we have actually contributed positively to the national debate on this. We curate, adapt training materials on open science and reproducibility. We do outreach in different disciplines and regions via our annual ambassador program, which starts like 15 to 20 people from different disciplines. We have people from like archaeology, geosciences, marine biology, education, you name it. I mean, this is quite diverse, and people actually are elected for a year to represent the network within their own communities. And we also do a lot of activism with open science partners in Brazil. Brazil has a very well-developed open science community, which was there much before we came in. And it's been a big pleasure to collaborate with these people in terms of like trying to get government and other entities to promote open and reproducible science. So yeah, this is it. So final message here. I do think that the organization thing matters here. I think improving research requires building structures that make better use of the human potential within academic research. And I think we have only collectively explored like a very tiny fraction of this design space. I mean, we have experimented with this decentralized national collaborations, but this is just like one of the very, very many ways to do things. And I think we should be bolder and in developing and testing new approaches to organize scientific efforts and make better use of the human capital within academic research. Thank you very much. I should be thanking the coordinated team of the initiative. The Scientific Institute funded this from the start and funded the labs. I mean, I've been like complaining about the labs all the time. You might think that I don't like them, but it's really been like a super privileged experience to work with. I mean, really like the big learning years actually to have this very privileged window to like glimpse of people working in real life. So I very much deeply thank the labs involved, and like this is their reward as well as mine. Thank you.
There will be questions, and congratulations. I mean, especially for not just, I mean, for taking this beyond just blaming and crying foul because we have 30% reproducibility rate, but really an impressive analysis and also so many things that take this hopefully to stages where things get better.
One question or idea, make it sense to combine both projects. This should be for me a good tool to increase your reproducibility. So I think we should make together.
Yeah, we have not worked with like large scale genomic data as he has, but I do think that this kind of like systematic quality control. We can be much better at having these things within pipelines. We have like very simple experiments, but still like people do quality control in very particular haphazard ways. So like I think standardized ways to like do quality control in techniques that a lot of people use is super helpful.
Very much the whole setup around, right? So what you described to talking all these people and having these connections with them. So like the vision that I mentioned to have like this giant circular experiment, right, would need exactly that. So
yeah,
I will approach you.
And if you need bad quality, they do have a lot of that. That's not a problem.
Alistair.
Has anybody tried to sort of standardize the labs themselves in terms of the time of day you do things, the air conditioning, temperature, the physical conditions in the lab?
Yeah.
If you were Henry Ford, you'd make sure that they're the same.
Yeah, but there's a downside to that as well. Instead of like, yeah, if some things only work like in the afternoon, if you tell everybody to work in the morning, that will kill off possibly an effect. I think other replication projects, like other not replication projects. I mean, there have been attempts in. I know some in animal science, some in cell line experiments to try to like very much standardized procedures across labs. You can do it. I think it works. We here made a deliberate attempt not to do it because like if you over standardize to something that's actually not the original condition, it might be unfair against the replication. I mean, if you actually don't know what time of day the original experiment was done, I mean, it was better to let every lab decide on that and like, yeah, let's do it in the morning, because if the actual experiment was at night, you might end up having non-reproducible conditions just because you standardize something differently. So we tried to not to standardize everything that was not known about the original paper. So like we did not make an effort to standardize. I think you can go the other direction. Like how much can we standardize this? I think you can go, and I think, I mean, judging from other projects, I think that goes some way. I mean, I think it depends a little bit on the kind of experiment as well. I think like cell line experiments are easier to standardize if you take like some data from the behavioral field. I mean, even if you do a very big effort into the standardizing procedures, still you get some things that are just like, yeah, different breeding conditions for the animals for a long time, and like you end up with different things anyway. So I mean, it's hard to standardize, and I'm not sure it's always desirable. I think you can actually do over standardize sometimes, like lose something that's important.
There's a tension between standardization and generalization. Evident.
Wonder whether there were typical obstacles when you started out to overcome in your community. I can think of two. That people said, why should I participate in something that does damage to the reputation of our field? And why should I sacrifice my time for participating in the replication effort? So if those two obstacles were there, how did you overcome them?
I think we actually did not get that much of a bidding in terms of that. I think the project had actually very like good publicity in general. I think we made a very explicit effort not to care about the original results. It was like we're doing this meta science thing, but like we're not really judging this. Actually, I mean, if you want to look up what the original studies were, that you can, but it's like in a supplementary table somewhere. So like we're not trying to like. We're not particularly interested in like judging other people's work. We always took this first person kind of approach. So I think we largely managed to like avoid being seen as like the replication police or something. We did get some criticism in terms of like why look at Brazilian science? I mean, it just like makes like Brazil look bad. We're already like we have a lot of problems. Like why we're focusing on this and not on the international community that has the same problems probably. My answer is like I'm trying to focus on what I can change. So like this is a reality I kind of like work in and where I can see I'm making a difference. But that one we got. In terms of like wanting to participate, we're very surprised by like how many people actually wanted to participate in this. We were like when we opened up registration, I think we're aiming for like 30 to 50 labs. We got like 70 something in the first call only. I think we may have been lucky. I think like our funder has very good visibility within Brazil. They helped a lot in the outreach. Brazil is very big on social media, so like we did there was like peak Facebook time, and like we actually did a lot of outreach via social media. And I think we got lucky in terms of like we started out the project with like private funding within a time like Brazil was like in a very bad financial crisis in terms of research. So like a lot of people were just like they did not have a lot of money to do stuff. Like if somebody appears like, please do this, we'll pay for it. Maybe just people were up for it because there was those were hard times.
Maybe this is the time also for Germany to start. Christopher, and then
I wonder the more you are going to tackle the human factor and standardization, there could be two serious adverse events. The first is that you get rid of the fun factor in science, which is very important for productivity to some extent, and also the issue of serendipity. So finding something more or less by accident, which can come out of problem in the experimental conduct in the first place, but then you're able to replicate and find something new. So how are you approaching this cultural aspect?
I fully agree. I don't think we should get rid of either of the fun factor of like we should keep exploratory research going. I think it's just a matter of like and it's fine. It's probably fine that most research happens that way. I think I think it's a good way to like actually, draw on the creativity of people. And it's just sometimes you have to confirm stuff, and I'm not sure we actually have the structure to confirm. So when we really need to, of course, you don't. And the other thing, I mean, you don't have to apply the super rigorous standards to like every single scientific project. It becomes unmanageable, right? I mean, so I think most science can go on as a kind of, perhaps with a little bit of improvement, but like most science can go on in this much kind of like free-form thing. But I think like every scientific field should like, at some point, like every year, like, okay, this is the fifth, like 10 or 15 or 20 findings that we really want to be sure about and be able to confirm this. And I'm not sure we have the structures for confirming this. I'm not sure what the ideal, I mean, should have like a one center that this is, just dedicates itself to replication. We have very established consortium that can do it, but I think we do have to create these structures for confirmatory science. I don't think they have to be the mainstream or the majority of the scientific endeavor. I think it's a particular thing, but I think it's important that it gets done at some point.
You were telling us about the researchers were able to predict whether the finding would actually be replicable. I mean the researchers of the original experiments or the researchers actually conducting the replications?
It's an independent. We had 70 people participating in the prediction project. Some of them were within our labs, some of them were not. So like, and they had to predict a particular method. So I think it's like 20-something people predicting the outcome of the experiments for each method. And there was a survey we did. We ran a prediction market as well. Actually, the survey worked better. I think perhaps people did not understand the market as well as we wanted. But it does. I mean, people vastly overestimated replicability on average. Like, So like we had 25%. So we asked them about statistical significance. So that's just 20% of experience replicating, 80% not. People were more about trying to. They usually said something like 50% or something. But all of the experiments that were successfully replicated were like above average in terms of like what people thought they were likely to reproduce. So like the ones that people did not find likely to reproduce, like they actually did not reproduce. So I mean, there seems to be some accuracy in at least predicting the ones that are like not likely to reproduce.
Do you think there could be some potential to use that information, like actually in publishing?
Probably, I think, but that's very tacit, right? So like psychology has the same. I mean, this doesn't matter actually for psychology experiments. I think predictions, markets, and survey to go. Even lay people can actually predict the outcome of psychology experiments to some degree. So I do think there's a lot of tacit expertise that is not super. And I mean, we're now analyzing what people used as justifications for the prediction. So like we had an open question like why are you predicting this? And they said, the effect size is large, or this is very significant, or like this is easy to do, or whatever. We're now analyzing these answers, but we're just starting. So, but there seems to be useful information in terms of like how people can predict. I was surprised that this actually worked. I thought it would be hard in biology because most people are like not experts in the field. They're just like predicting. You're working in like immunology and predicting some neuroscience result. I thought but it actually worked a little bit. So it's interesting too.
I think we have to move on. Actually, by the way, in the science that I come from and Alistair knows about, if you predict 100% that it will not replicate, you will be right in 100%. But that's maybe a particular problem. So congratulations again, and we are moving. Actually, I should also mention that there will be a small reception afterwards. So if you had a question and didn't have a chance, it will be possible later on. So we are coming to the final category, to the individual award, which is awarded with 150,000 euros. And it's my particular pleasure now to welcome the person who took the initiative to submit the nomination. Because he believed the winner's work and efforts truly deserve this recognition. So please welcome Richard Lucas. He's a professor of psychology at Michigan State University, and he will say a few words about our winner.
So I'm really happy to be here to introduce Dr. Simine Vazire, the winner of this year's Einstein Foundation Individual Award. Simine is a professor in the School of Psychological Sciences at the University of Melbourne. And in preparing this introduction, I thought back to what I think is the first time that I met Simine. And if my memory is correct about this, it was in 2007 at the annual meeting of the Society for Experimental Social Psychology. Now, neither Simine nor I are experimental social psychologists, so we gravitated towards each other as like-minded personality psychologists in an unfamiliar environment. But what was most notable about our meeting was the topic of our conversation. So for those of you who know about the history of the so-called replication crisis or the credibility revolution in psychology, we often trace the beginning of this shift in perspective to around 2011, when a number of really critical papers came out that led us to question some of the findings in the field. Yet almost 5 years before that, Simine and I talked almost exclusively about what would later be called questionable research practices, research degrees of freedom, and p-hacking. And although we lacked the terminology and did not yet understand all the mechanics by which these problematic practices affected the quality of the science that was produced, Simine already had her eyes on the issues that would ultimately win her this award. Now, over almost 2 decades later, 2 decades since that conversation, Simine has accomplished at least 2 major achievements that make her a very deserving recipient of this award. And she's really become a leader in these areas. So first, from very early on in her career, she focused on editing. She served as associate editor for at least 5 different journals and was editor-in-chief at an important social psychology journal during the immediate aftermath of the replication crisis. Simine was, and this is in the context of and constraints of an academic society that was quite conservative on these issues at that point, yet she was progressive in the policy changes and really served as and provided a model for those who wish to shift their own editorial policies to promote research quality. She was also the co-founding senior editor and later editor-in-chief at Collabora Psychology, which is an open access journal that was designed to be at the cutting edge of what can be done to improve the openness, transparency, and quality of the research that is published. And now, remarkably, given all the resistance she faced in trying to implement these policies, Simine's now the editor-in-chief of Psychological Science, which is the top empirical outlet in psychology. At that journal, she's instituted wide-ranging changes that I think many would not have thought possible at a mainstream journal. And so she's again serving as a model for what others should do in the field. Now, the other thing I think that Simine did throughout her career working on these issues, and again, she's done this with great success, is to advocate for improved research practices throughout the sciences. In addition to shifting her academic research and writing to focus more on meta-science and research quality, Simine also made this work very accessible to those within and outside the field. She authored a widely read blog about these issues. She's hosted a successful podcast about them. She even co-founded an entire society, the Society for the Improvement of Psychological Science, which is dedicated to improving research quality. And I think what's most impressive about this and what is Simine's ability to persuade people about the importance of these issues and the utility of adopting these new practices that address them. Not only does she have an impressive grasp of the nature of the problems that scientists face, she's remarkably effective at being able to discuss and explain these issues to those who may be on the fence or even on the other side of the fence from her own perspective on these issues. She tackles these conversations with confidence and assertiveness when needed, humility when appropriate, and for those who have read her blog or listened to her podcast, she often does it with a great sense of humor. So it's been impressive to watch her over these years, and I think she's had a tremendous impact on how psychology and other sciences have responded to the challenges of the replication crisis. So congratulations to Simine for this well-deserved award. Please welcome Dr. Simine Vazire.
Thanks so much. Thank you for that introduction, Rich. It's great to be here. What I'm going to talk about today is some of the lessons that I've taken away from my field's replication crisis and then our reform movement and revolution since then. And so this is a little bit of a confessional of kind of my growth since 20 years ago. And also, I'm hoping that there's a lot that we can learn from each other in different fields so that other fields don't have to make the same mistakes that we made. And we've learned a lot in psychology from biology, medicine, and other fields that have gone through this as well. So the first lesson I learned, it's kind of embarrassing that I had to learn this, but it's that transparency is actually pretty important. So if you go back 20 years, if someone had asked me if I would share the data that I used to publish a paper, I would have said no. In fact, I did say no in some cases. And that was the norm in my field, was that, no, you're not going to share your data. And it's pretty embarrassing when you think about it, because if you went to buy a car and the seller said, no, you can't look under the hood, we would say that that's crazy, right? So why should scientists have any less integrity than a car salesperson? So I came around, and my field eventually came around to understanding that, of course, if you're going to ask someone to buy your result, they should be allowed to look under the hood. And actually, this analogy works pretty well, because there's a paper in economics that won the Nobel Prize by George Akerlof, using exactly the used car market to make this point, that if there isn't transparency, actually public trust in that market it'll be a vicious cycle. So it's actually a very simple argument. If there's information asymmetry between the seller and the buyer, in this case, the researcher selling their work and the reader or community buying the work, then and the buyer knows that. They know that there's information the seller's withholding. Maybe we know that when we read a scientific paper, there were more studies done or more analyses done than what is reported. Only the best ones are reported. Then we're in a situation of quality uncertainty. We know that we can't judge the high quality products from the low quality products. There's no incentive for sellers to sell high quality products, because they can pass off low quality products for high ones. So they show that in such a market, the quality of the products goes lower and lower, the buyers have less and less confidence in the market, it leads to a vicious downward cycle, and trust falls. So I made the same argument that if it's true in used car sales markets, it's also true in science. And there's just really no excuse for scientists to say, no, you can't look under the hood. And if we do that, we risk losing public trust in science. So that was one hard battle to fight within our field of psychology. But we did, I think, generally win that battle, that it's now understood that transparency is important. We can't just say, no, I don't want to do it. That's not a good enough reason. But even then, we learned that transparency is really hard to change. So in psychology and social sciences in general, we've had quite a few initiatives, mostly grassroots initiatives, mostly led by early career researchers, to try to increase transparency in research practices and in publication practices. And this has been going on for 15 years or so in psychology. I mean, actually, the open science movement obviously goes back way further than that. But a kind of really concentrated effort in psychology started with the replication crisis. And Tom Hardwick, whose work I'm going to be talking a lot about today, has done some great research tracking the prevalence of open research practices in psychology. And what I'm going to show today is just a piece of that, looking at kind of the early days before these reforms had really taken hold, and then more recent numbers in psychology for a couple of open practices. So first is the open sharing of data. So Tom's group coded how often published articles in psychology have a link to open data. That's all. Not even like whether what's there is actually the right data, or whether it's interoperable or reproducible or anything like that. But just by that standard, back in 2015 or so, it was 1.6% of articles, random sample of articles in psychology. And by 2022, it was 15%, and in a prominent sample, 16%. So it went up by an order of magnitude. So it's a pretty big change, but it's still in absolute numbers really quite low. So that's with a huge amount of effort, a huge amount of activism, and a lot of change, at least subjectively within the field. It really feels like a lot has changed, but we still have a long way to go. Another transparent practice that we've been pushing a lot in psychology is pre-registration. And so here we had 2% of articles in psychology had a pre-registration associated with them in 2015 or so, and now it's maybe 10%, depending on whether you focus on a random sample or sample of the more high-impact journals. So again, a long way to go. Probably the ideal rate of pre-registration is not 100%, but I think it's arguably quite a bit higher than the current rate. So one lesson from this is that we are making a lot of progress on transparency, but it's not easy. So to borrow Brian Nozick's pyramid of culture change, which I think is a useful way to think about how to change scientific cultures as well, with transparency in psychology, I would say we're at the stage where we've made it possible. There's now resources and so on. I think that it's rewarding even in many cases. There's incentives to do so. But that leads me to my next lesson, which is one that I learned as a journal editor. I think that there's a lot of really good reasons to move slowly up this ladder. We don't want to jump too fast, but there's also really good reason to not go too slow. So my next lesson, and this came from my experience as a journal editor, is that we really should move as fast as we can responsibly towards requiring transparency. And I put an asterisk there because, of course, there should always be exceptions for when transparency is not in the best interest of science. But for the most part, in psychology, in many cases, what's in the best interest of science is for research to be transparently reported. And what happens if we stay in this middle ground where transparency is maybe asked for or nudged towards, but not required, is that the incentives will always favor those who opt out. And this is a situation I find myself in over and over again as a journal editor. So imagine that you're asked to evaluate 2 manuscripts. In one manuscript, the researchers reported really transparently. So maybe you can see the batch effects. I don't know if I'm using that term right. Or maybe you can tell that there was a lot of flexibility in the data analysis and the result really isn't robust to alternative specifications. The other paper, there's not enough transparency. You can't tell. So all you know is they're reporting one result and it looks really, really good, but you don't know how many other things they tried. So you have 2 options as a journal editor or as a reviewer or as a reader of those papers, right? You can either believe the second paper, give them the benefit of the doubt, in which case you're punishing the first paper. They could have written that polished, nice looking paper. They chose not to. Or you can not believe the second paper and say, no, I don't believe you. You're not letting me see enough. You're not letting me look under the hood. And let me tell you, that doesn't make you a very popular journal editor. So really the only solution is to say, well, we're going to penalize those who aren't transparent. We're going to give a boost to those who are. But really, ultimately, I think the goal is to move to where we have a level playing field and transparency is expected of everybody. So we did that at the journal that I'm now editor-in-chief of, where Tom Hardwick is senior editor for statistics, transparency, and rigor. So we don't require pre-registration, but we do require open sharing of data, code, materials, and we do a computational reproducibility check. And again, we allow for exceptions when we deem that that's what's in the best interest of science. So that was a lot of work to get there. It took 15 years or so of psychology's replication crisis and reform movement. And even that is really not enough, right? Transparency is not magic. And actually, Tom's own work has shown some of this. So he looked several years ago at a psychology journal that was a really early adopter of requiring open data. And so he looked at a handful, about 35 articles that had been published in that journal after they required open data. So the authors chose to submit there, knowing they would have to share their data. They shared their data. So really early adopters of open practices, probably a best case scenario for really good transparent practices. And he tried to reproduce the results of those 35 papers. And what he found was in about a third of the cases, he could reproduce every single result in the paper himself or his team could. In another third, they could do so after getting assistance from the original authors. And in about a third of cases, they weren't able to reproduce every result, even though the data were open and even with author assistance from the original authors. So this shows that transparency definitely helps, right? Without transparency, we just wouldn't know about these problems, but it's not enough. And so transparency helps bring the problems to light. And then whether you improve as a field or not really depends on what you do next. So there's 2 possible paths if your field increases transparency. One is that we detect errors, we correct them, we learn from those mistakes, and we increase in credibility. But that's not the only possibility. The other possibility is that transparency leads to showing that we're making the same errors over and over again, and people keep finding them, and we're not learning, and then we get lower credibility, right? So transparency doesn't guarantee credibility. Transparency guarantees that we'll get the credibility we deserve, which is still much, much better than not being transparent, right? I put an asterisk there because actually transparency is not enough, right? So being transparent means it's possible that someone could look under the hood, but you really want to make sure as a field that if you're going to be transparent, you also make sure that there are people going in and looking under the hood and fixing any problems that are there. Otherwise, you're going to end up in that bottom path of losing credibility. So we call this organized skepticism, and borrowing a phrase from Merton, the sociologist of science. And this is actually doing the work of detecting errors and fixing them and learning from them. So as hard as transparency was to change in our field, and we have made some progress, what I think is going to be even harder is organized skepticism, actually detecting errors, actually fixing them and learning from them. And here I would say that psychology is still at the bottom of this pyramid, where it's barely even possible to do that work. It's certainly not rewarding at this point in our field. And that's where we're at now, I think, with our reform movement, our revolution is trying to improve that while we continue to try to improve transparency. So where is organized skepticism supposed to happen? Well, one place, I think, if you ask both the public and scientists, is they would say that peer review is a part of organized skepticism, right? And that brings me to my next lesson from psychology, which is journals are part of the problem. So we would hope that journals are helping to identify errors to make papers better and so on. And just to pick a random time that I heard someone express this, it was a few months ago, you hear all the time, for good reason, this is a reasonable expectation, right? We typically expect that errors will be caught in the peer review process. And one of the things that psychology's replication crisis brought to light was that, oh no, a lot of errors are making it through the peer review process, right? So you might have heard of the Reproducibility Project Psychology that came out in 2015. So this was one of the early flashpoints of psychology's replication crisis, where the open science collaboration tried to collect new data and rerun 100 studies published in top psychology journals. So these are journals with rejection rates of 90% or more for the most part. They were picking out, the very best psychology findings, and most of them failed to replicate in this replication project. But it's not just irreplicability, it's all kinds of other problems. Once we started to look under the hood, we found many different kinds of errors. So just to take a particularly embarrassing one, our journals were publishing articles where the test statistic and degrees of freedom did not match the p-value. Very often, these mistakes are happening. And in one of 8 psychology articles published in a few decades, in 8 top journals, those inconsistencies were what are called gross inconsistencies, where one part of the statistic would suggest that the result is not statistically significant, but the p-value was reported as statistically significant, or rarely vice versa. So we aren't doing a very good job at journals of doing error detection. And this is captured, and I'm going to pick a little bit on one particular editor-in-chief, but I think she can take it. She's editor-in-chief of Nature, who was quoted in the Wall Street Journal as saying, "I would not want to think of my peer reviewers on the papers as some kind of police squad catching mistakes." And look, this is just her being honest, right? Peer reviewers, there's a reviewer crisis. It's not realistic to think that they're going to be there recomputing statistics or checking pre-registrations or something like that. But there's a discrepancy between what we ask peer review to do, what we kind of talk about it as doing, and what it actually does. And that led me to write an article in Wired called "Peer-Reviewed Scientific Journals Don't Really Do Their Job," which makes it even more surprising that they let me become editor-in-chief of Psychology's flagship journal. But this isn't really just a criticism of journals and editors, it's also a criticism of what we expect of them, right? It can't be the case. Journals are never going to catch all the errors. Still, they could do a better job, right? I think that we should expect more from journals and from peer review. So, 1 in 8 articles in psychology having a gross inconsistency in statistics, now that's easy to fix because Michelle Naughton and her colleagues developed StatCheck. So now we can run psychology papers through that and identify any inconsistencies in the statistics. But one thing that shocked me about psychology's replication crisis is how little people expect of journals. So if you remember the Reproducibility Project Psychology that I talked about, they targeted 3 specific journals in psychology, 3 very prestigious psychology journals. And I've given talks to psychology audiences where I asked them, can you name any of those 3 journals? And they're like, don't remember, because that's not where the reputation stuck, right? The reputation for the failures stuck to the field. Maybe it stuck to a few high-profile findings, but the journals got off scot-free. And it's not really clear why, right? These were journals, again, that were rejecting 90+% of submissions. And it turns out that a lot of what they were publishing was not easy to replicate. One of those journals is the journal that I'm editor-in-chief of now, Psychological Science. So we took that responsibility seriously. We think the field should be asking, what are you going to do in light of this? We found a problem, so what are you going to do now? So we've implemented some checks along the way, in addition to the traditional external peer review. Again, this is Tom Hardwick doing a lot of this. I apologize, the fonts are messed up on my slides. But the details don't really matter, but basically we have this team of statistical and transparency and rigor editors, as well as a repeat network of volunteers who help us do transparency checks, computational reproducibility checks, and so on. So that now when something gets through the peer review process, in addition to having experts in the content peer review the work, we also have experts in statistics and transparency checking the things that are relatively easy to check. So it was interesting coming into this journal and trying to kind of earn the prestige that the journal already had. Because one of the things that's frustrated me over the years is that journal prestige is so sticky, right? That journals have it and they keep it, and it doesn't really matter what they do or don't do. But I think we should come to expect more of our journals, and I think journals can do more than they're already doing. And basically, we should withhold prestige from journals unless they live up to some, again, they can't be perfect at detecting all errors, but they could do more than they currently do. But even if journals did do a better job, right, there's always going to be some errors that make it through. And our goal should not be to make 0 errors. That would be way too conservative of a way to produce and publish science. So even peer review is not magic, even if it's done really well. So we need critics. We need people who go in and do post-publication critique of the work that we think is the most solid. And perhaps the darkest time of the replication crisis in psychology for me was the reaction to critics. So we had some early critics, we had some people who ran replication studies, who pointed out errors, et cetera. And in my field, I'm sad to say that the reception of those early critics was quite negative. So they were called things like replication bullies, human scum, methodological terrorists, second stringers, destructive iconoclasts, and my favorite one, which because of the formatting error is now crossed out, but is accuracy fetishists. This was launched at the Reformers as if it was an insult, but I think it's actually worn as a badge of pride. So there was a lot going on around the time that the problems were getting aired, and this kind of goes to the question that was asked earlier in the Q&A, that yes, there was resistance of saying, why are you airing our dirty laundry in public? Why are you making us look bad? And so on. There was a very high profile person in the field who wrote a column called A Call to Change Scientific's Culture of Shaming. This was not a call to be nicer to the critics. This was a call for the critics to stop being so critical. So in her column, she wrote, to be sure, constructive critics have a role with their rebuttals and letters to the editor, but those should be subject to editorial oversight and peer review. So basically critics go through the appropriate channels and let the gatekeepers gatekeep. So are journals doing that? Are they encouraging letters to the editor and commentaries? Are those getting through? So we worked together with Annie Wamond, who did a paper looking at psychology journals and their policies, and whether they have an article type for post-publication critique. And the vast majority either explicitly or implicitly did not. So this is no, and this is yes. So something like 35% did have an avenue for that. Then we looked at how often are there actual post-publication critiques published in journals. And we randomly sampled 200 articles and found one instance of post-publication critique. So journals are not really providing much of an avenue, or maybe there's just not enough incentive for people to do it. We're not really sure. So one of the things we did at Psychological Science when we took over was to kind of invite our critics to come and be part of the team and help us detect the errors before we publish them. So this is almost all of the statistics, transparency, and rigor editors at the journal, and some of whom are in the room. And one thing I like to point out is that one of the ways that we chose who to recruit for this team was who had published pretty harsh and correct criticism of our journal in the past. So there were a few. So Tom Hardwick himself had done meta-research on computational reproducibility of articles in the journal. Sophia Cruel had led another project. Similarly, the Data Collada trio had identified cases of fraud published in the journal. And so we brought those people, if they were willing to come on the team, we're like, let's find those errors before we publish them. And actually, maybe the best life advice I have, am I old enough to be giving life advice, is when you find someone who's a really good critic of your work, hire them. That's the best thing you can do. And then maybe it's weird to call this a bright spot in psychology's replication crisis, but actually I think it was really heartwarming, was when this trio got sued for identifying fraud. When Francesca Gino sued them, this was a scary moment for psychology's reform movement because it really had a chilling effect. People are much more afraid now of getting sued if they point out errors. But the lawsuit was dismissed. And maybe the most heartwarming thing was when we did a GoFundMe to support those researchers, the number of people who contributed was really stunning. So for me, it was a really clear marker, this was in 2023, of how far we'd come since 2011 in the early days of the replication crisis. My next lesson is one that I think I still really struggle with and I don't know how to deal with. And I think I see other fields going through this, which is that a crisis can look a lot like normal science. It's really, really hard to tell the difference, partly because of all those sources of variation that Olavo talked about, that there's, random error, there's true heterogeneity, then there's bias and questionable research practices, and there's fraud. And so I was on a study committee of the National Academy of Sciences in the US, where we were tasked with writing on the state of reproducibility and replicability in science. And in one of the early chapters, there was this sentence: the advent of new scientific knowledge that displaces or reframes previous knowledge should not be interpreted as weakness in science. This is absolutely true, of course. Of course, it's completely normal and healthy in science for new knowledge to displace old knowledge, something we thought we knew, turns out not to be the case. But what's scary about this is that how do we know what's normal, and how do we know when we're just like going down the wrong road over and over again, hitting a dead end and not changing course? And in psychology, it felt a little bit like we were at risk of doing that. And it can be really easy to fool ourselves that this is just part of normal science. So we might end up like the dog in this cartoon, but we don't often see how the cartoon ends, but it doesn't end well. We don't want to be these people. So how do we make sure that we're not actually in a crisis, that it really is just normal amounts of error and correction and so on? We have to look for signs that we are engaging in self-correction, learning from our mistakes and so on. But it's really hard to know what self-correction should look like. And I turned to philosophy of science to look for answers of like, what makes science self-correcting? What does it actually look like? And so Naomi Oreskes, who's a historian of science, has a book called Why Trust Science? And I got really excited when she was leading up to like, okay, so how do we know that science has self-correcting mechanisms? And she says, we know that the same way we know other empirical facts through observation. And she said, when we observe scientists, we find that they have developed a variety of practices for vetting knowledge, for identifying problems and attempting to correct them. And there was, to be fair to her, there was a little bit more specific examples in this, like peer review was one thing she named, but it was still to me a lot of hand waving. Like I didn't find a really concrete, like, here's how you would know, and here's how you would know if it's not self-correcting. I'm going to put up another quote, not by a philosopher, but by someone you might have heard of, James Heathers, who I think is kind of an amateur philosopher in his social media posts. So this was a tweet he wrote in 2019, not a response to Naomi Oreskes, just happened to be the same year, where he wrote, in reality, the mechanisms to correct bad science are slow, unreliably enforced, capricious, run with only the barest nod towards formal policy, confer almost no reward, and sometimes punitive elements for a complainant who might use them. And this is more consistent with my experience, even being on the other side. As the editor who should be, working to correct mistakes that we've published, I now see how hard it is to do that, how many systems are in place that make it very, very difficult to correct, even when we know there's an error. So I think that we really should be asking ourselves, are we actually doing the work to make our field, our discipline, self-correcting? Otherwise, I don't think we can say this is just normal science, right? We have to actually be able to point to something. So this is where the organized skepticism comes in. So I think kind of going to Olavo's point about the balance between kind of exploratory and confirmatory work, you could also ask about the balance between, advancing discovery, going at a fast pace, and actually stopping and checking that we're not making too many errors. So in psychology, for example, we have a group called the Psychological Science Accelerator. I think it's great. They're doing really great things. And actually, they're not about going fast and breaking things or anything like that. But I'm just playing on their name, really, to make this point that just as much as we should invest in discovery, we also need to invest in verification, correction, and so on. So maybe we should have a well-funded psychological science garage where we bring our findings if we want to make sure that they're really up to the task of building on them, making public policy off of them, and so on. And we're starting to see some institutionalized groups like this. So we have the Institute for Replication, which started off as a group that did reproducibility checks of findings in economics and political science, but they've started adding psychology to their repertoire. So that's my story of the crisis. I have one more lesson, that the thing that is kind of my obsession right now, because often when your field finds itself in crisis, you really want to find a lifeboat and jump on that lifeboat. And I'm afraid that the next lifeboat that we're going to jump on is meta-analysis. And so my last lesson is meta-analysis is not magic, right? We thought transparency would save us. We thought peer review would save us. We thought post-publication critique would save us. And we say, okay, well, even if all those things fail, we still have meta-analysis and it's at the top of the evidence pyramid, right? So we're going to be fine. But it's not magic, right? If the things lower down in the evidence pyramid are not solid, meta-analysis is not going to save you. And there's been some excellent meta-research in the social sciences. My favorite is this paper comparing meta-analyses and large-scale pre-registered multi-lab replication projects like the one that Olavo was talking about. This is in social sciences. So they looked at findings where there was both a published meta-analysis and a published really large-scale replication effort. And they found that when they compared that across a dozen or so different findings, the meta-analytic effect sizes were almost 3 times as large as the replication effect sizes. In my opinion, the replication effect sizes are much more trustworthy and unbiased. So this is quite concerning. And then if you say this to psychologists, they might say, okay, yeah, but we have ways to detect bias in meta-analysis and to correct for it. So they tried that too. They implemented 3 methods for correcting meta-analysis for bias, but these methods did not substantively improve the meta-analytic results. So I think that if we're hoping that evidence synthesis or meta-analysis is going to save us, it's not. We really need to fix the foundations of the field. So I'll leave it there. Sorry again for the formatting, but the first lessons are about transparency, we need to have transparency, we need to have better peer review and post-publication critique, and then a few more lessons. And I'll end on another tweet by James Heathers. Science is self-correcting, sure, when we correct it, not because of magical progress. And I want to thank the people who have supported me through all this. And yeah, this award really, it shouldn't be called an individual award because, as we know, no career is ever an individual's doing. So thank you to all the people who helped and supported me and inspired me. Thank you.
Someone tried to turn on the light. I've tried here and I don't know how it works. So maybe. Thank you very much. I'm sure there are questions.
Yeah, do you have any suggestion for other fields that might not be as far in dealing with the replication process? You know, it affects a lot of different fields, and my impression is that the fields have stared at different points. It's how openly these problems are being addressed.
Yeah, I mean, I can say what I think helped in psychology's case. So one was kind of really going back to fundamental values and making sure that we all agree what are the values of being scientific, right? So in the beginning of the replication crisis in psychology, there were people arguing against transparency, and very often you could just kind of go take a step back and say, okay, but what do we think makes something scientific, right? We learned in 3rd grade that scientists show their work. So isn't that a fundamental, like, do we agree on that in general scientists should show their work and so on, and then kind of work to find where the disagreement is. Very often you can get really good agreement on these abstract principles and values, and then using those to kind of build your argument for the more concrete practices. I think that helped in psychology. My sense was that it didn't change anyone's minds who was like really established, but it helped reshape the next generation. They heard those arguments, and I think those were pretty convincing. So I think appealing to really principles and values can be quite helpful. Also just finding other like-minded people. So one thing in the early days of psychology replication crisis, many of us who kind of felt like we saw the emperor has no clothes were the only one in our department or our city or whatever. And so luckily we had social media at the time. I think maybe it was better than it is now. I don't know. But at conferences, organizing, hackathons or even just social events to meet up and work together and support each other, I think that can be really important. And then once you have the grassroots, trying to institutionalize it. So we formed a society, but you could also, do reproducibility group or other kinds of networks now that exist. Connect with if your country does have a reproducibility network, like the networks that Olavo talked about, you could connect with them. Now there is a bit more, I think, kind of transdisciplinary infrastructure for open science and things like that. So trying to plug into those.
Is it possible that the different patent rights cause the lack of transparency? You have the discussion now on artificial intelligence, and here you have methods, and sometimes even methods have patents. Is this possible?
Maybe in other fields. I think it's not in psychology. I don't know of many methods in psychology that are patented, but yeah, that could be very relevant in other fields. In psychology, honestly, the arguments against transparency were open.
Method, even ideas. Ideas. This is a problem to not describe the ideas. So.
Oh, yeah. I don't think so. I think the equivalent in psychology might be that sometimes we collaborate with corporations to get data. So if you need data from Facebook or from insurance companies or from whatever, then we can't share the data because those companies have a self-interest in not, competitive, wanting to keep their competitive advantage and so on.
I wonder whether there are disciplines that are better and who are also engaged in experiments, and whether there's something to learn from. I have the idea that astrophysics is somewhat better than psychology or biomedical research, but is this in the nature of their subject, or are they better as a community?
I was sure that physics was better, and then I started getting invited to physics groups to think about psychology. That honestly was very scary for me. I was like, wait, what? So I don't know anymore, but I mean, certainly there's certain very specific practices like blinded data analysis that astrophysicists engage in that we should be doing more widely, or perturbing data, things like that. So I think it's really interesting how different subdisciplines even have developed specific solutions. I remember talking to someone in spinal cord injury research in like 2012, and they had already discovered p-hacking. They called it testing to a foregone conclusion. I was like, oh, that would have been useful to have that concept earlier. So I do think there's like pockets of things that are generalizable across disciplines that we can learn from each other, but they're fairly isolated, I think. Yeah.
Yves Moreau, and this gives me the opportunity to introduce Yves Moreau. We have the 2023 winner of the individual award with us. So, Yves.
Yeah. So, what about the social incentives? Because this is kind of fueling the whole thing. So, how could those be addressed better? So, first, there's better practices, and I think we all want to do that. Behind that, there is this push of publishing more, having more sexy results. That is a big driver.
Yeah, I mean, I think that's really hard. I think that's maybe a reason that psychology was vulnerable to this crisis, was that people like the topics that we study, so we can get book deals, we can give TED Talks, so it kind of amplifies the already problematic incentives even within academia. We had even more of that by having these opportunities outside of academia. I don't know really, I know I don't have a magical solution to them, other than I think that we really want to fix it with carrots. You know, we give people badges or we give them gold stars for doing the right thing. I don't think we can fix it only with carrots. I think there have to be sticks, because the incentives are so strong, right? If you can claim that you've made some big discovery that's going to help people in their relationships, I mean, everyone's going to want to hear about it. You're going to get so many interviews, you're going to get book deals, you're going to get all this stuff. So unless there's some kind of disincentive that, no, if you're saying this in the name of science, I mean, it's one thing for a guru to say that, but for a scientist with their scientist hat on to say, I have found the solution, it's going to prevent divorce, it's going to prevent breakup or whatever, that's irresponsible. And so if there's no negative consequence, the kind of benefits of that are going to get out of control. There's some interesting modeling work by Carl Bergstrom and Kevin Zollman and a few different groups showing that actually, like, even something like fraud, if you look at the actual chances of getting caught, and of course we have to make some guesses here, and the penalties for getting caught compared to the benefits, it's really hard to make sense of why everyone isn't committing fraud all the time. So I do think we need to not only increase the incentives for good practices, but probably have more disincentives for bad practices somehow.
Yeah, so toward the end of your talk, I was expecting the rule number 10. But then there wasn't.
Just to have a round number.
So I wondered, I thought you would say something about the role of AI in the whole process. So where do you see it? Is it on the bad side or on the bright side?
Well, I think I don't, so I should say I don't have enough expertise to make an informed judgment. All I'll say is that even the bad side might have a silver lining, in that I think AI is just making really, well, not just, but one of the things it's doing is making really obvious the problems that were already there, so that peer review is falling apart. It's not, we already had a reviewer crisis. Now we're going to have all this AI slop to vet in addition to the human slop. We need a different system. We need to make our peer review system much, much stronger somehow. But even without AI, we were having a problem, but I think AI is helping to accelerate that problem in a way that maybe will force us to deal with it more. Maybe AI will also be part of the solution. So I do think a lot of the stuff like that, a lot of the more tedious aspects of peer review, you know, stat check type things, we ideally could automate. So I think AI will help. I think AI will exacerbate the problem as well. I don't know which effect will be stronger, but I would say that even without AI, we needed an overhaul to our system. So if we can do that.
I think it's not crazy to think that a severe crisis can really help. I mean, I think psychology, if you take the Netherlands, for example, and what Diederik Stapel actually did for this country. I mean, he should get a medal, because now we have a country that is really.
Yeah, he had a few helpers also. There were like a trio of fraud cases in the Netherlands in psychology.
Yeah, so I think I subscribe to all of your 9 points, and that's super. I'm totally aligned. So, but sometimes when I ask myself also, all these practices, you said it's easy to do, but some of them are not so easy. Some will have substantial resources required to open up responsibly data sets, for example. To do replications, for example, is substantial, like Olavo showed. How many years did you do this now? 10 years, 15 years? So, but Tim Erring, it's that, and also the last thing that Tim just did. I mean, it's a huge effort, and we know something then about if a certain field, how good is the replication. But how will you balance, and what are your thoughts on the balance between transparency and also the efficiency of science in itself? progressing.
But I think the progress, when we were progressing really fast because we weren't slowing down to do replication, that was the illusion of progress.
Yeah,
So I actually
totally subscribe to
yeah.
this, but now we're kind of.
But I think, well, what I think we had to do in psychology is stop being addicted to the illusion of progress. Like, I think that if you compare, okay, yes, we could have, 1,000 new discoveries a year and get on the cover of Time magazine and this and that. But if half of that isn't true, or the other choice is to have, 10% of that much success and discovery and all that, but 90% of it is true. And we know which 90%, okay, maybe that's the ratios are wrong there, but I think that we have to remember that the really fast-paced discovery was largely an illusion. And what's even scarier is that, let's say even half of it was true, that's great. But the fact that we can't tell which half was true, which half wasn't, means all of it is now under suspicion. So that's not useful progress, right? We really want is to have high confidence in the findings that are, getting a lot of attention and traction and so on. So I don't think that there's any choice in the matter. Of course, there's details we have to work out. Yeah, what proportion of investment should be in verification work versus discovery, for example. But I think that's a problem for future us. I think right now we just need to get above zero, right? So I think there's like in psychology in the early days, there was a lot of hand-wringing about, oh, are you saying everybody should do replication? Are you saying 50% of journal articles should be replications? No. Let's get to 1% and then we can start arguing about what the ideal rate should be. But we have barely gotten off the ground.
Maybe a final question that I have. How's your journal doing since you're editor?
I think fine.
Okay. Yeah. Do you see any response in terms of? You must with this vetting of papers, pre-vetting.
Yeah.
You must have increased your rejection rate. Before sending stuff into review. Or did you?
No, actually, not really. No. So our acceptance rate is the same. It takes longer.
Okay.
Yeah.
And
but
so no
my
complaints.
best estimate of our acceptance. Oh, I'm sure there are complaints. No complaints to my face.
Okay. Okay. So thank you very much. Congratulations again. So we're coming to the end of this little symposium, and this is an invitation to all of you. Now that you know what this award is about and what kind of people are being awarded, what kind of projects. Obviously, we are trying and hoping to also get beyond psychology and biomedicine. But the call for the 26 award is out, and there's, I think, plenty of time still with the deadline. So if you have ideas yourself, or if you know someone who you think should be considered, should be nominated, nominate, spread the word, contact us if you're in doubt whether this would make sense. So thank you very much.